The project was titled RNA sequencing using the Seurat package in R to study distal lung cellular plasticity. I was the mentee. My name is Sawhney, and my mentor was Dr. Crystal Markinet. First, I'm going to go over some background of the study. You can go to the next slide. The distal lung is an intricate organ that is responsible for important physiological processes such as alveologenesis, which is the final stage of lung maturation, and it's also responsible for repair after injury. A key characteristic of the distal cell is its cellular plasticity, particularly when it comes to alveolar epithelial cells. Previous studies have identified the primary stem progenitors in the distal lung as alveolar epithelial cells, specifically the type 2 cells. So I'll refer to them as AT2 cells. Through transient and intermediate states, these AT2 cells undergo cell fate regulation, and understanding these cellular transitions has important applications to things like tissue repair and regeneration, which I will probably mention multiple times throughout this presentation. There is previous precedent related to studying the dynamics of the distal lung using single-cell RNA sequencing. The significant transcriptional states of lung epithelial cells have already been studied, relating to their roles in lung development, homeostasis, response to injury. What still remains unclear, however, is the specific relationship between these AT2 cells and the known epithelial cell types. So my goal in this study, with the context of the background that I just mentioned, was to annotate the transient intermediate cell states of the AT2 cells in the distal lung using publicly available single-cell RNA sequencing data. By using the Seurat package in R for reanalysis, I aim to gain a comprehensive understanding of the dynamic shifts in cellular identity that occurred during a critical period of lung organoid culture. So now I'm going to go over the methods that I used and the results that I derived from them. The data that I analyzed in my paper had already been published and analyzed in a 2020 Science article by Choi et al. So I first wanted to give some context on how they obtained the data that I ended up using. The researchers used mice, which were exposed to tamoxifen and then either PBS or bleomycin. The tamoxifen they used was used in order to enable greater gene control, and then the PBS was used in control groups, and it just formed as a form of homeostasis. They used it instead of water so that the cells didn't rupture or shrivel up. The bleomycin was used in the injury-induced group of mice because it's used to induce inflammation, fibrosis in mammal models in the lungs for a short period of time. The data in the primary study was taken from the mice on day 0, and then they were isolated for single-cell RNA sequencing analyses on day 14 and day 28 of sitting in organoid culture. For the purposes of the study that I did, we only used data from day 0 and day 14. To study the organoid data from Choi et al., we performed a few initial analyses, the first of these being mitochondrial contamination. And I have a couple bullets on this slide, but I really talk about it in the next slide with the figures. As shown in these figures, mitochondrial contamination varied across cell types, indicating to me that there was an issue with the difference in the quality among the cells. In order to visualize the effect of mitochondrial contamination on the different cell types present within the organoid data, I plotted the percent mitochondrial contamination versus the cell type as it was annotated by the Human Lung Cell Atlas, as shown in the first figure. And the second figure was able to confirm for me that the source of the difference in the variation that I was seeing was due to cell type rather than the day that the cell was analyzed, so the cell sample. The next initial analysis that I conducted was a principal component analysis, or a PCA. I noted 2 main sources of variation, which is shown on the graph in the next slides. These sources can either be biological, which is an actual biological difference in the way that the cells were cultured, or a technical difference or variation, which is due to something that may have happened in the lab. So that could be like a random increase in temperature or something else that changed in terms of like technicalities. So data in the first figure shows us that the first principal component was the largest source of variation between the two cell samples, and the genes that are identified as most variable in this first principal component were associated with known differences between epithelial and fibroblast cells. Given the fact that our sample started off with AT2 cells, which were epithelial cells, and the fact that fibroblasts are often used as feeder cells in culture settings, I was able to determine that this variation was likely a biological variation involving the fact that the day zero cell sample was not grown in organoid culture at all, whereas the day 14 cell sample spent 14 days in that organoid culture. Looking at the second figure, I noted that PC2, the second principal component, represented ciliated cell markers. And so when I observed gene signatures that were known to be enriched in ciliated cells in the PC2 plot, I was able to kind of get a hint to a small cluster of cells that was not presented by the primary analysis of the data in that Choi et al. Paper. And this was something new that I noticed. Next, I used jackstraw plotting to determine which principal component analyses were significant using their standard deviations. The first figure shows, the first figure shows us the jackstraw plot. So on the right side of that first figure, we can see the p-values of each principal component. And then on the jackstraw plot, it plotted each of these p-values, and any point that lies above that linear trend that you see indicated a significant principal component. As shown in the second figure, the standard deviations between the observed and expected values of the first two dots on the plot, so the first two dots like towards the left of the plot, indicated that these were our two main sources of variation, and then the remaining 6 dots on the plot were smaller sources of variation. After doing these initial analyses, I moved on to observing the data in a more unique way. I used UMAP to identify and visualize the cell clusters, which were numbered 0 through 6, and I also observed each of their properties. The first figure pictured here identifies where each of the clusters are. So based on, I don't know if you can see the legend that clearly, but based on the color in the legend, it shows us kind of where the cell clusters were identified. It's this slide. Sorry. Comparison between the first and second figures was able to demonstrate that the cell clusters 0 and 5 constituted the majority of day 0 cells, and the cell clusters 1, 2, 3, 4, and 6 were presented in the day 14 cell sample. So the second figure shows the 2 different cell samples on the 2 different days, and then by comparing it with the first figure, which shows the different cell clusters, we can see which clusters appeared on what day. All cell clusters from both samples came from surfactant protein C, which is shown in the third figure, which we already knew. The analysis shown in the fourth figure identified 3 main distinct cell types within the samples, type 1 cells, type 2 cells, and fibroblasts. It was previously known that type 1 and type 2 cells were majorities within the cell samples. Like I said, those were the epithelial cells that we saw. And then again, like I said, fibroblasts likely arose from the way in which the organoids were cultured, so possibly as feeder cells. Both the fifth and sixth figures display measures of the quality of the cell clusters. The fourth figure indicates a lower number of genes in the fibroblasts as compared to the type 1 and type 2 cells, which again was expected. And the mitochondrial contamination in the fifth figure confirms that each of the cell clusters compared to each other had reasonable percentages of mitochondrial contamination. The organoid data from the surfactant protein C mice was then annotated with lung map data obtained from the publicly available website that's called Azimuth's Lung Cell Cards. So SFTPC is a known marker for type 2 cells, which is pictured in the first figure. HOPX is a known marker for type 1 cells, which is pictured in the second figure. And then the third figure shows all the different types of cells that were part of the annotation. So when we compare which cell clusters contain the marker for type 1 cells and which cell clusters contain the marker for type 2 cells, And then we compare it to the third figure. What we can obtain from these figures is that known marker genes for type 1 and 2 cell types were enriched in the populations, and these are annotated on those graphs as either AT1 or AT2. So overall, as our kind of final results area, a comparison was made to see if the annotation made with the organoid data was validated by an independent annotation database. So that was where our lung map cell cards was used. On the next slide, we could see a table, and data in this table confirms that my analysis was validated. My annotation indicated 3 main distinct cell types, AT1, AT2, and fibroblasts. Additionally, my analysis claimed that a significant number of AT2 cells would shift into AT1 cells over 14 days in culture. These findings were confirmed by the comparison. However, there are certain differences that I noted. One being that there was a significant shift of 112 fibroblast cells to Tufts cells, 418 fibroblast cells to SCMF cells, and 84 fibroblast cells to mesothelial cells. And then there was also a shift of 254 AT1 cells to B cells that was also noted, and I touched on all of this in my discussion. Yeah, so now I'm going to talk about my discussion. Primary analysis of the data used in this paper presented by Choi et al. So this was the primary analysis, not my analysis, and it suggested 2 different findings. First, that there were 4 distinct populations of type 2 cells that were identified, and it was just the type 2 cells. And second, a DATP transitional cell state was also identified. My findings show that while the analysis of the same data in my paper also observed a DATP transitional cell state, my reanalysis of the data showed many other epithelial cell types that were not described in Choi et al., so not just those type 2 cells. By using newer ways of reanalyzing the data and identifying the epithelial cell states, I was able to identify type 1 cells that the original paper was not able to identify. Other unexpected findings, such as the shift of fibroblast cells to different cell types, suggested technical variations in the data, given that fibroblasts don't typically shift to epithelial cells. And similarly, the shift of AT1 cells to B cells also suggested a technical variation. This was most likely misannotation errors that may have occurred, given that epithelial cells don't typically shift to B cells. The significance of this research lies in its potential to manipulate cellular transitions for therapeutic purposes. So understanding the molecular mechanisms that underlie these type 2 cell fate decisions and transitions could lead to strategies for enhancing lung tissue repair. The findings also emphasize the importance of 3-dimensional organoid models in capturing the complexity of the lung microenvironment. Future studies should delve into the molecular cues and environmental factors that drive the transitions that we observe, exploring the impact of the extracellular matrix, mechanical forces, and signaling networks could probably provide a more comprehensive understanding of lung epithelial cell plasticity. And finally, in conclusion, as an overview, in this study, we were able to successfully annotate transient and immediate cell states of AT2 cells during lung organoid culture, and our analysis revealed dynamic shifts in cellular identity, suggesting remarkable plasticity of the lung and of the distal lung structure. Validating our findings with publicly available lung map data confirmed the presence of AT1 and AT2 cell populations. Unexpected cellular fate shifts were able to underscore the complexity of the microenvironment's influence and the different things that can happen. And moving forward, further investigation into the molecular mechanisms driving these transitions could pave the way for interventions with lung regeneration and lung disease. Finally, thank you first to Talaria for giving me the opportunity to connect with Professor and help me learn a lot of new things. I don't think I would have been able to get this opportunity in an easier way. It was really fun to work with Dr. Markinette, and she was always available to kind of talk to me when I had questions. I've never done something like bioinformatics before, so it was really interesting to learn this from her, and I was really glad to have this experience. And then on the slides next to this, it's just my references for my paper. Thank you.